Hi everyone, my name is Nafisa Jadavji, and I'm an assistant professor at Midwestern University. I'm located at the Glendale campus in Arizona, and today I'm going to be presenting some data from my lab. I started my research laboratory in July 2019, and I present some data that was collected by some graduate and medical students. So the title of my talk today is Does Nutrition Impact Ischemic Stroke Outcome? The Role of Dietary Vitamin B12 Deficiency on Ischemic Stroke in a Mouse Model. So like I said before, my lab investigates specifically neurological diseases. We're interested in diseases of aging, specifically the neurovasculature, and related mechanisms. So we focus on stroke as well as dementia, and then maternal nutrition in terms of how nutrition during pregnancy and lactation impact offspring neurological health after birth. So we're currently funded by the American Heart Association, or we have funding from the American Heart Association, and we also have had funding from the Arizona Alzheimer's Consortium, and some of that data I'll be presenting today. You can learn more about my research group and our accomplishments and what we're working on by visiting my lab website. So today I'm going to talk a little bit about, or give you a bit of background on nutrition, as well as stroke. In terms of nutrition, I'll be focusing on 1-carbon metabolism. And then I'm going to go through some experimental data from my research group looking at vitamin B12 deficiency and stroke. So let's start at the beginning and we'll talk about 1-carbon metabolism. So 1-carbon metabolism is a pathway, a metabolism pathway in our body. It plays important roles in a number of different aspects. So I'm going to walk you through those. And that, first of all, is nucleotide synthesis, specifically the synthesis of purines, as well as DNA repair, so the removal of uracil, and neurotransmitter synthesis, the synthesis of acetylcholine, so this neurotransmitter both in the central nervous system as well as in the peripheral nervous system. 1-carbon metabolism generates methyl groups that are involved in membrane lipid metabolism, as well as in the generation of acetylmethionine, or SAM, which is a global methyl donor. And in the brain, it plays an important role in neurotransmitter synthesis. These methyl groups also help remethylate homocysteine to methionine. And homocysteine, we're going to talk a little bit later about that. High levels of homocysteine in blood of humans or in patients has been shown to be associated with a number of negative health outcomes. So for example, increased risk for cardiovascular disease. So this is 1-carbon metabolism in the cell. It's a little daunting in terms of all the pathways and different components, but I really want you to focus on the nucleotide synthesis that I brought up earlier, the methylation, so this generation of acetylmethionine, and transsulfuration also, which we didn't really talk about before, but this is a component that I wanted to highlight also of 1-carbon metabolism. So the big players in 1-carbon metabolism are folic acid, or folates. So folic acid, rings a bell. If you're a woman or, a man who's had babies, in the sense that women of childbearing age are recommended to take adequate levels of folic acid in order to promote the closure of the neural tube in the developing baby or embryo, in the sense that the neural tube forms the spinal cord and the brain. And another player in 1-carbon metabolism is vitamin B12, which we're going to talk a lot about today. And I'm going to present some interesting data on, as well as choline, which is a nutrient. So this is 1-carbon metabolism as a whole. You can see all the nitty-gritty here. And why is it important? Well, we know, like I've outlined before, the important role that folate and choline, components of 1-carbon metabolism, play in the cells: nucleotide synthesis, DNA repair, methylation. We also reduce, as we age, our ability to absorb nutrients decreases. And so this is also another reason why one-carbon metabolism is important, because the drivers in one-carbon metabolism are things that we get from our diet. And increased levels of homocysteine, again, the whole idea that high levels of homocysteine lead to negative health outcomes, such as higher risk for cardiovascular disease. Also, there are changes in our body as we age, so changes in our stomach, pH, or acidity, in our intestine, that sometimes change, that do change how nutrients are absorbed. And so that's specific really to vitamin B12 in this talk. And this prevalence can, be somewhere between 5 to 40% of the population. And it's really dependent on the marker that's used for deficiency and the cutoff. So vitamin B12 deficiency can be marked by increased levels of homocysteine or changes in levels of methylmalonic acid. So this is how vitamin B12 deficiency is confirmed in clinical populations. And we see that the elderly population is really a population that has these changes in vitamin B12 deficiency. So this is a figure that Gillian Yan, a previous graduate student in my lab, a master's student in my lab, put together. So we get vitamin B12 primarily from animal sources in our diet. It is then, broken down in the stomach. Vitamin B12 is also referred to as cobalamin. And so with acids in our stomach, it's broken down and then moved into the intestine for absorption. And so this is how vitamin B12 is brought into from our diet into our body. And so what happens in the aging process is that our stomach acids change. And so there is this change in the breaking down of vitamin B12. And what also happens is that there are changes in levels of something called intrinsic factor, or IF, which helps with the absorption of vitamin B12. And so that's why we see this high prevalence of vitamin B12 deficiency in older individuals. So we're going to switch gears here and we're going to talk a little bit about stroke, and I want to introduce you into the topic of stroke. So stroke is where there is a change or a reduction of blood flow to an area of the brain. There's two different types of stroke: ischemic stroke and hemorrhagic stroke. So hemorrhagic stroke is when a blood vessel ruptures in the brain. So blood doesn't reach a target and there's this buildup of pressure. And then there's also ischemic stroke. Ischemic stroke is another type of stroke, and there is a thrombi that forms within the blood vessel. This then results in reduced blood flow, so reduced oxygen, reduced glucose going to cells. And so then, so cells like neurons, different types of glia cells that, require this constant flow of oxygen and glucose to function. So then there becomes cell death, and so we see this brown tissue here. So this is tissue that and cells that have died, that then has led to this tissue loss. So in the US, stroke is one of the leading causes of death. There's also a higher prevalence globally, internationally, of ischemic strokes over hemorrhagic strokes. And stroke is one of the leading causes of long-term disability in terms of loss of function. And what we see in older populations, so people over the age of 65, is that there's mobility issues that occur after stroke. This is a graph from the American Heart Association from 2015 showing the prevalence of stroke by age and sex. So we see different age groups here on the x-axis, and you can see both in men and women, the number of strokes increase in the elderly population, in the 80 plus. However, we are seeing a change or a shift in the prevalence of stroke in younger populations due to the increased risk factors, the prevalence of these risk factors increasing. So things like hypertension, diabetes, obesity are all increasing in our populations throughout the world. And those are all risk factors for stroke. So we are seeing a change or a shift, which is not outlined in this graph. And I just wanted to bring that to everyone's attention. So what's really cool about the brain, or one of the cool things about the brain, is that the circuits are crossed. So here we have a stick figure. If this individual were to have a stroke on their left side of their brain, we would see impairments on the right side of their body because of the brain circuits being crossed. So if their stroke was in the motor cortex, we would see motor impairments on the right side of the body. So maybe issues with walking, reaching, and different motor functions. So that's something to keep in mind as I move along in my talk and talk about stroke outcome and how we assess that in my lab. So I want to get right into the data because that's the good stuff, and talk a bit about our rationale. So in the clinical population, there's a lot of data that associates high vitamin B12 deficiency with worse stroke outcome. So the work that my graduate student Gillian Yan did for her master's thesis, the aim of it was to understand the role of a vitamin B12 deficiency in ischemic stroke outcome using a mouse model. So in this study, we used both males and females, and we made them vitamin B12 deficient. And we also had a control diet. And our hypothesis was that we would see worse stroke outcome in the vitamin B12 deficient animals compared to the controls, potentially because of more apoptosis occurring within the brain, within the ischemic region. So what did we do? Well, this is our experimental timeline. We hit 10-month-old animals. We put them on a control or vitamin B12 deficient diet for 4 weeks. We then induced stroke. We waited 4 weeks, and then we did some stroke outcome measurements. So we have 2 components to stroke outcome measurements in my lab. The first is looking at behavioral function. So we target something called the sensory motor cortex when we do an ischemic stroke. So then we can look at motor function. And then after we do motor function assessment, or we measure that, we then look at tissue. So brain tissue, plasma, and liver, other areas of the body to try and understand what's happening with the deficiency, with the stroke. So this is what we did in this study. In terms of our dietary manipulations, we had 0 levels of vitamin B12 in our deficient diet, and our control diet had 0.025. We induced stroke using the photothrombosis model. So this involves an interperitoneal injection, or an IP injection, of a photosensitive dye called rose bengal. This then gets into the circulation, and then we use a laser to activate this dye. And then what that activation results in is that there is this thrombi formation that occurs in the blood vessels that are targeted with the laser. And so we get oxidative stress and all of these things occurring to cause this thrombi formation. And so we put the animals into a stereotax, and we target specifically the sensory motor cortex, which is shown here. In this crescent violet stained image, you can see damage about 5 weeks after we've induced ischemic stroke. And so this is what we do to induce ischemic stroke, and we do a unilateral damage. So what that means is we damage only one hemisphere of the brain, so that then we're able to assess one side of the body and look at the impaired function versus the non-impaired function. So again, highlighting this brain circuits being crossed. So we do assess some behavioral function, like I mentioned. We use the accelerating rotorod task, which measures coordination and balance, and the forepaw placement task is a task we used here to look at forepaw placement. So we have an impaired forepaw and we have a non-impaired forepaw. So we're able to compare the usage of this in animals that have an ischemic stroke. So what did we find? Well, we measured levels of homocysteine in both plasma and in liver. And again, we used males and females. And what we saw in plasma with it was that there were increased levels of homocysteine in the plasma of these animals, of the vitamin B12 deficient animals, as well as in liver. So this confirms that we then had our vitamin B12 deficient diet worked. We also then looked at the amount of damage in brain tissue. So Gilly sectioned brain tissue. She stained it for crescent violet. So you can see the staining here, and you can see this damage region here. And so what we were able to do is quantify it using image J, because we took coronal sections throughout the damaged area. And we're able to see no changes in ischemic damage between the different groups. What we did next is we went into the damaged region using immunofluorescence experiments to try and understand if there were any changes in apoptosis. So we used active caspase 3 to assess apoptosis in these animals, and we saw increased levels of apoptosis in the male deficient animals, the vitamin B12 deficient animals. And you can see representative stains here. So this led us to believe that something was happening in terms of cell viability. And so we wanted to look at survival of cells. So we looked at a marker called PAKT or phospho-AKT, and we co-stained sections with neuronal nuclei or NUN, as well as DAPI, which is a nuclear stain. So you can see representative images here. And what we're seeing is we're seeing more survival of neurons in the vitamin B12 deficient group, suggesting potentially that there might be some compensation occurring. And then what we also wanted to measure functional outcomes. So we used, again, these behavioral tests, so the accelerating rotarod. And what we see is that the male vitamin B12 deficient mice had impaired coordination and balance after ischemic stroke. So they stayed on the rotarod less. So as it was getting faster, they were not able to stay on it, and they were not able to achieve a higher speed of the rotarod while they were on it. And so we see this impaired coordination and balance in the male animals. And when we look at forepaw placement, we saw this reduction in forepaw usage in the male animals. So what I've shown you in this study is that we have increased levels of homocysteine in both liver and plasma in vitamin B12 deficient animals. Within the brain tissue, we see that there's really no difference between the ischemic damage, but we see more apoptosis within the ischemic region and neuronal survival, neuronal survival. And we see worse outcome in terms of coordination, balance, and reduced forepaw usage in the vitamin B12 deficient animals. So the data that we've generated so far confirms what we see in the clinical population is that vitamin B12 deficiency results in worse stroke outcome. And the males are more impaired compared to the vitamin B12 deficient males. And that what we're adding to the literature with this work is that the mechanisms driving this increased vulnerability or outcome after stroke are increased apoptosis, and that there is some sort of compensatory mechanism of neuronal survival within the damaged tissue. Now, we did this work in adult animals, so we're really interested in seeing what's happening in the aging model systems. So again, we used mice, but we used aged mice, and we specifically focused on female animals because we were interested in the female response to the ischemic stroke and stroke outcome, as well as the vitamin B12 deficiency. So our experimental design was similar. The only thing that was really different is that we used a lot older mice. They were 18 months, so they model aspects of an aged human. So what we saw in terms of our results was that these animals, the vitamin B12 deficient females, had more damage. So their ischemic region in their brain was larger. We saw more apoptosis. Specifically, we looked at neuronal apoptosis, and so we saw more of that within the damaged region. We also measured mitochondrial metabolites because of the link of mitochondria to apoptosis. And so we did significance testing and enrichment analysis, and so that's what I'm showing you here. So we are seeing changes between, so this is an ischemic and non-ischemic brain tissue, and we're not seeing a change between the ischemic and non-ischemic tissue. Where we're seeing changes in is in response to the diet. So we're seeing changes in metabolites, mitochondria metabolites that are involved in the TCA cycle, reductions in those. We're also seeing changes in when we did the pathway enrichment analysis and the phenylalanine, tyrosine, and tryptophan biosynthesis pathways. Next, what we did is we measured levels of metabolites within the cecum tissue. So the cecum is what connects the small intestine to the large intestine. And so we were interested to see if this diet changed and stroke changed metabolites there. And what we do see is we see that there's a dietary change in methylmalonic acid as well as creatine. And methylmalonic acid, like I mentioned before, is in response to, is usually measured to confirm vitamin B12 deficiencies. We also did a fold change analysis, and we show that there are metabolites where we see changes. So specifically DOPA, 3-aminobutyric acid. So these metabolites involved in neurotransmitter synthesis as well as other metabolites. So we also assessed stroke outcome, functional outcomes. So we used a neurodeficit scoring scale. This is the vertical screen task. So the animals are put on their cage lid and we look at whether they're able to stay on, because the screen, the cage lid is turned perpendicular at a 90-degree angle. And so we look at whether they're able to stay on, whether they move up, they move down, or they fall off. And so we see a higher severity score. So the vitamin B12 deficient animals perform worse on this, this component of the neurodeficit score. We also looked at the accelerating rotorod, and we see the vitamin B12 deficient animals aren't able to stay on as long, and that they take longer to cross the ladder beam. So the ladder beam task is a horizontal ladder which the animals walk along. We look at the number of errors they make, how they place, and the time it takes them to cross. And where we saw a difference was in the time. The animals took a lot longer to cross the horizontal ladder. So what I've shown you is that in this set of experiments is that we have vitamin B12 deficient females that have more damage in the brain. They have more apoptosis. We see changes in mitochondrial metabolites, as well as in cecum metabolites, and we see functional changes in terms of worse stroke outcome. So again, this experiment combined with the previous experiment, we're seeing that a vitamin B12 deficiency impacts motor function outcome after stroke. It reduces it. It also changes metabolites in the brain, in the cecum, levels of apoptosis. So we're adding these mechanistic data to the literature in terms of understanding what's happening with vitamin B12 deficiency, and then contributing that to the data, to the literature, and how vitamin B12 deficiency changes the body and the brain. So in terms of the study limitations, we used a model system. So we use mice. We also use a photothrombosis model of stroke. There are several model systems of stroke. So there's the middle cerebral artery occlusion, there's the endothelium 1 model of stroke, the photothrombosis model. Now each model has its pros and its cons. The photothrombosis model is something that's well tolerated in animals, so it's easier to do on older animals, and we don't have a high mortality. We see nice damage regions consistently in all animals. We also are able to really look at functional outcome after these lesions because we target such a specific area of the brain. There are some cons in terms of this model, the penumbra, as well as the reperfusion component of stroke. So when blood flow is restored back to the brain, what happens to that? But there has been some data that's showing changes in cerebral blood flow after photothrombosis. So there is, some data coming out, suggesting that there might be a reperfusion component to photothrombosis. Our work was really to try, or has started to try to understand the mechanisms through which a vitamin B12 deficiency really impacts stroke outcome. And then this data that we've generated and that I shared with you today is really, leads it to the idea of combination therapies for stroke-affected patients. So combining a pharmaceutical with some sort of lifestyle change. So, nutrition, in our case, seems to be, might be an ideal candidate for this combination therapy. There are several people I'd like to thank or acknowledge. Gillian Yan did a lot of the work that I presented today, as well as a collaborator, Heiwei Gu, and his graduate student, Panez, did a lot of the metabolite work or the analysis. Joshua Poole did the apoptosis analysis in the aged animals. I'd also like to thank the Arizona Alzheimer's Consortium for funding some of the work that I presented today. And I'd always like to thank the animals for having them and for being able to use them to try and understand what's happening in the brain and in the body in response to stroke and nutritional changes. Thank you all for your time in listening to this talk, and I look forward to all of your questions. I would also like to thank Ben for the opportunity to present my work. It's always a really nice fun time to put data together and put talks together, and we're in the process of writing some of these papers up, so it's really nice to think about this data, and I really look forward to your questions. Thank you. Take care. Bye-bye.